Now I have the pleasure of introducing you to Professor Stacey Harmer. She's actually had some of our students in the past work with her as interns doing research, and she's going to be telling you about circadian rhythms in plants. Probably something you haven't thought too much about. So it's very, very exciting. I'm glad to have you here once again. And she's also incredibly friendly, and feel free to ask any questions.
Yeah, absolutely. I really enjoy getting questions while I'm, during my talk. So please don't hesitate to just raise your hand and ask. It kind of makes it more interesting for me, and I hope it makes it more interesting for you as well. Joe, do you still like people to give a little bit of their background before they start? Okay, so I've been at Davis now for about 7 years, so that's exciting. I just got tenure last year, and that's as you probably know, that's a big step. So before that, I did a postdoctoral fellowship in San Diego at Scripps Research, and that's where I started working on the topic I'll tell you about today, plant circadian rhythms. Before that, I worked in animal systems. I did my PhD at UC San Francisco, where I was studying something actually not too unrelated to the previous talk you heard. I was studying how B cells, which are the antibody-producing cells in animals, how they respond to pathogens or to antigens. So you may wonder, and before that I did my undergraduate work at Berkeley, so I got my bachelor's degree there. So you may wonder how I went from studying mammalian B cell signal transduction to studying circadian rhythms in plants, and topically they are pretty different. I switched for a couple of reasons. One was that I wanted to be able to do forward genetics. Have you guys heard much about using forward genetics in your other classes or in this class? No? So one of the beautiful things about genetics is it allows you to understand the basis for pretty much any trait that you're interested in. If you're interested in understanding how eye coloring comes about in fruit flies, you can do mutant screens and identify fruit flies with altered eye color, and then you can isolate the gene responsible for that mutant phenotype. So you don't have to know anything about the mechanism underlying a process. All you have to do is have a good genetic screen, and I'll talk a little bit about what a screen is later. So I wanted to be able to study processes that we didn't really understand anything about. So working in mice wasn't so great. Mice are expensive. They have a kind of a long life cycle, and it's just hard to do forward genetics. And plus, I didn't really want to have to sacrifice a lot of mice. That's actually why a lot of people end up in plant biology. They're not so crazy about killing a lot of animals for their research. And I was interested in circadian rhythms because I think it's a really interesting and mysterious process. How is it that animals, or plants for that matter, are able to keep track of time? So are you guys familiar with the term circadian rhythms? PR, yeah. So have any of you had jet lag? Yeah. So you know that when you cross time zones and your body clock is out of sync with the environment, you just feel terrible. And so it turns out that many organisms have circadian clocks. Plants have clocks, some fungi have clocks, even some bacteria have circadian clocks. And all of them use these internal clocks to sort of tune their biology with the changing environment. Okay, so any questions about my background before I talk about our research? Okay, so this is going to be very heavy on the bio part and a little bit light on the photon part. So we study the circadian clock in this one plant called Arabidopsis thaliana, which is sort of the fruit fly of the plant world. It's a very easy plant to work with. And this shows a flower clock that was designed by Linnaeus. And he had noted that different species of flowers open their flowers at different times of day. And so he imagined a garden where you could tell the time of day by which flowers were open. So the 4 o'clock plants open their flowers in the afternoon, and some of these night blooming plants that are pollinated by moths, open their flowers at night. And so the idea was you could just look at your garden and tell the time of day. And actually some people have planted these. I don't think Linnaeus ever did. So, but I like this example because it illustrates that one process, in this case the control of floral opening, can be differently regulated in different species so that it occurs at different times of day. So for some flowers that are bee-pollinated, it's advantageous if they're open during the day, whereas moth-pollinated flowers are open at night. And you can see right there, I think, how important to the biology of the plant it is that they can tell what time of day it is. In this case, so that they can coordinate reproduction with when the pollinators are around. So I'll have 3 parts to my talk. I'll give you an introduction to circadian rhythms. I'll talk about using luciferase as a reporter. So that's a bioluminescent molecule you've probably heard about already. And if I have time, I'll talk about a research project where we found that a hormone pathway in plants is regulated by the circadian clock. Okay. So for plants and for other organisms as well, we can generalize and say that there's an internal clock or oscillator, and it generates 24-hour rhythms. And what's I find fascinating is that in all the organisms that have been looked at, rats, fruit flies, bacteria, plants, these oscillators are cell autonomous. That means you can isolate single cells, and they still have a 24-hour clock running in those isolated cells. So that's, to me, a really fascinating biological question. How can a single cell keep track of time? And so that's in common to many different types of organisms, but of course clock outputs are going to be different. So animals would have their activity is regulated by the circadian clock, your sleep-wake cycles. If other animals, we have our blood pressure is regulated by a circadian clock, so that's why many heart attacks occur in the morning. That's when your blood pressure is naturally at the highest. And if you're in terms of plants, I'll talk about different kinds of outputs that are influenced by the clock in plants. So the hallmark of a circadian rhythm is that if you have the organism in isolation, so insulated from all environmental cues, such as changes in temperature or light, you still see 24-hour rhythms in the output. So, for example, there were some famous experiments where scientists took college students who were volunteers, and they got them to go into underground bunkers, and they put them in isolated them, gave them constant light, constant temperature, and then just measured their activity rhythms. And they found that they had a roughly 24-hour rhythm to their sleep-wake cycle. So that's what I mean by this oscillator is not dependent upon changes in the environment. So their clocks didn't stop without input from the world. But of course, our clocks are sensitive to the environment. That's why you get over jet lag. You cross several time zones, and then you get the new light cycles and temperature cycles, and social cues will help reset your clock.
I'm just wondering, do they still have a clock if they're not exposed to the environment in the first place?
Okay, yeah, great question. You can't do that experiment with people, obviously, but it has been done for fruit flies. So fruit flies have certain behaviors that are clock regulated, and there was one experiment where they bred the fruit flies in constant darkness for 800 generations, and when they got to the 800th generation, they still found that those flies had a clock, a circadian clock that ran with a roughly 24-hour rhythm. So if you look in things like cave fish, that have been, in some cases, isolated from changes in the environment for, who knows how long, some of those cave fish still have a functional clock, but some of them don't. They've probably lost it just through chance. If a mutation occurred in their clock to break it, if there's no environmental selection, then it won't be maintained. Okay. So this general scheme is true for all the organisms that have been studied, but the actual components of the clock are different between different species. So the fly and the human genes are very similar, but they seem to be totally different from the plant genes. So we think they may have evolved separately. So this is my example of circadian rhythms in a variety of organisms. Here's someone suffering from jet lag. You can measure 24-hour rhythms in activity in rodents. If you put a wheel in a cage, the mice run on that wheel with a 24-hour rhythmicity. They're active at some times of day and inactive at others. If you're studying neurospora, which is bread yeast, bread mold, you can see a 24-hour rhythm in a developmental process. And if you're studying plants, this is a plant grown in constant light, and a little video image was taken. You can see a 24-hour rhythm in leaf movement. So the leaves go up and down with the 24-hour rhythm. And if you have a plant that's mutant for clock genes, that rhythm is going to be altered. All right, and so actually, this rhythm in leaf movement was one of the first clock outputs noticed. So about 2,000 years ago, a Greek wrote up in his log books that he noticed these 24-hour rhythms in leaf movement. And then Charles Darwin actually wrote a book on the power of movement in plants, which it was helped with the sun. This was published in 1897. And he set up an experimental apparatus where he had a plant with a glass plate in front of it, and he had a grease pencil. And I guess he was ill at this time of his life, so he was mostly lying on the couch. But he would make these little marks on the sheet, on the glass pane, to indicate where the tip of the leaf was. And he could show that it went up and down with a regular pattern. And he concluded that the periodicity of those movements was inherited. So this is one of the first examples of someone systematically studying leaf movements. And indeed, he's absolutely right. If you change the genetics of the plant, you're going to change the rhythms of the leaf movement. All right, so why are these rhythms so common? Well, it's been pretty obvious. We live in a world with 24-hour rhythms in a lot of things. Light-dark cycles for one, temperature, water availability. You know, if plants care a lot about water availability, if you're an animal, availability of prey or activity of your predators, all those things change at different times of day. So it makes a lot of sense to have an internal clock that allows you to anticipate those changes so you can be ready for them when they come. And we also think that having a clock allows organisms to coordinate their metabolism, so things that should happen at the same time do, and things that are incompatible could be segregated by time. So I took this movie from a website called Plants in Motion, which has lots of great stop-action movies. So this is showing a 24-hour rhythm in petal opening for a tiger jaw plant. So I'm going to focus my talk on well, if I get to that part. But anyway, I hope that I'll be able to talk about circadian rhythms and how they affect plant movements. Okay, so this is to convince you, I hope, that circadian rhythms matter to plants. This is an experiment published 5 years ago now. And they did a very clever set of experiments. They took Columbia plants, which are a strain of Arabidopsis plants that is very commonly worked with, and they took mutants that other people had isolated, where the clocks ran faster or slower than a wild-type plant. So if you measured the time of leaves up to down to up again for a wild-type plant, that would have a roughly 24-hour time span. But this one mutant called TOC1 does that more quickly than wild-type. This is if you have the plant in a constant environment, so no environmental cues. So it's a short-period mutant, whereas GTL is a long-period mutant. So they did experiments with these three genotypes. And here they took wild-type plants and grew them in a 24-hour day, so 12 hours light, 12 hours dark, from the time they planted the seeds to when the plants matured, and it made a certain amount of biomass. Then they did the same thing, but they grew the plant in 10 hours light, 10 hours dark, and it made about 50% less biomass. And similarly, they did the same thing with 14-hour light, 14-hour dark cycle. So these plants all have gotten the same number of photons, right? They're able to carry out, ideally, or hypothetically, they'd be able to carry out the same amount of photosynthesis. And yet the plant grown in a 24-hour day fixes much more carbon than the other plants. And they were able to show that this had to do with the circadian clock. With this experiment here, they took the long period mutant CTL, grew it in a 20-hour day versus a 28-hour day, and it did much better in the 28-hour day. And conversely, the TOC1 mutant, which has a short period, grew much better in a 20-hour day than a 28-hour day. So this indicates that for these three different genotypes, having a clock that matches the environment enhances growth. Okay. Yeah?
What do you mean when you say it was grown in a 28-hour day? Is that what you mean?
Yeah, so it was cycles of 14 hours light, 14 hours dark. So all the way from when they planted the seeds to when they measured the plants. Other questions? Mm-hmm.
So now when they think about growing plants in outer space, are they actually looking for ones that can, they can be pointed at the sun all the time, which could be a problem. Are they trying to optimize or do experiments now to see, because I don't know if you can get more biomass with exposing them for a longer amount of time, if you have like mutants.
Yeah, so I think probably not over if you're over 6 weeks of time, probably these would be on average exposed to about the same number of photons. But there have been some studies I know with monkeys, for example, where they try to say how monkeys behave in a Martian day length, which is about 28 hours, not 24 hours. And they generally don't do as well, like their clock cannot entrain to that longer day. So I know there's research in in animals to try to see if you could come up with a drug or a treatment that would allow the animal to deal with a 28-hour day. Presumably, you could do the same thing with plants. Mm-hmm. Yeah.
But does like the circadian rhythm or clock vary by season? Because it's not always 12 hours like.
Yeah, absolutely. That's a great point. So, but day lengths are almost, well, it depends where you are. If you're at the equator, it's 12 hours. But everywhere else in the world, the day length varies. Actually, I don't have a chance. Wasn't going to talk about this today, but people, plants and animals can use their clocks to measure day length. So they look for coincidence between the time of day that their clock says it is and whether the lights are on or not, and then they can infer, okay, that's a short day. It must be spring or fall, or winter, or that's a long day. It must be summer. And plants use that as a cue to flower. So some plants, like if you've ever tried to grow spinach, you find that they start to bolt and make seeds early in the summer, and then you don't want to eat the leaves. They don't taste very good. So that's a long day plant. It's responding to the lengthening days by flowering, whereas poinsettias or short day plants, they flower when the days are getting shorter. So it's an environmental strategy for when they're going to make their seeds and try to reproduce.
So you could artificially make plants flower
Yeah.
by varying light.
Absolutely. In fact, that's what they do with poinsettias to get them all, flowering by Christmas. You'd have greenhouses where you'd control the day length. And then there's been some studies of organisms like in northern Alaska, where you get those basically days and days of sunlight in the summer, or in the winter, days and days of no light at all. And they've tried to study if animals have functioning clocks under those circumstances. And again, it seems like some do and some don't. Yes, I think that one of the reasons I find clocks fascinating is you can ask all these questions about interactions between the plant and the environment, and how is the clock useful? Okay. All right, so in plants, we know the clock regulates, or at least influences, many, many processes, and I certainly don't want to talk about all of these. I will mention the gene expression. I'm a molecular biologist by training, so I care about gene expression. And that's a process that's many genes are regulated by the clock, both in plants and other organisms as well. And hopefully I'll talk a little bit about clock regulation of hormone signaling. So our organism is Arabidopsis thaliana. It's a small plant. It's a member of the mustard family. And for a plant, it has a pretty short life cycle. It takes about 6 weeks to go from one seed to the progeny seed. It's small, easy to grow, and the genome was fully sequenced back in about 2000. So that gives us lots and lots of genomic and genetic tools to use. It's easy to transform, which means it's easy to introduce foreign genes into these plants. And I'll talk about using firefly luciferase as an introduced reporter gene. And it makes a lot of seeds, which is great for genetics. And also what's great is you can just take those dry seeds and put them in a drawer, and come back 5 years later and pull them out and do studies on them. So it's great for that standpoint as well. Oh, and I should also mention, it has no economic importance, but the things that we find out in Arabidopsis, we can translate to things like soybean and corn and tomatoes and so forth. It turns out a lot of the important processes are highly conserved between Arabidopsis and more agriculturally important plants. So it's just like the arguments about working in fruit flies or yeast, that you can then apply them to humans. Did you have a question? No? Okay. All right. So I know you guys know this, but I'll just put this up as a refresher. If we have a gene in the genome, this is YFG, your favorite gene, it's controlled by a regulatory region upstream of the gene. If a transcription factor binds to that promoter, it causes the production of RNA, which then is translated to make your favorite protein. But then, like, one nuance you maybe haven't heard about is that you can have negative transcription factors that bind to that regulatory region and turn off the gene. So this is a very important regulatory step for organisms, whether genes are turned on or not. That's what, for example, makes our heart cells different from our liver cells, is what genes are turned on and what genes are not turned on. And you can have sort of battles between different kinds of transcription factors, some saying be expressed and some saying be silent. So in eukaryotes, animals, plants, yeast, the clock seems to be made of feedback loops of transcription, translation. So we know of 3 important clock genes in plants. One of them is called TOC1. Its message and protein levels are clock regulated, so they show peak expression at the end of the subjective day. So even in a constant environment, you see this pattern. And it somehow, we don't quite understand how, promotes expression of these 2 genes, CCA1 and LHY. And these are both the mRNA and the proteins are clock regulated, so they have peak expression around subjective dawn, so 12 hours out of phase with TOC1. And they inhibit the expression of TOC1. They directly bind to the TOC1 promoter and shut it off. So you have this kind of antiphasic relationship between the 2. And if you mutate any one of these genes, you disrupt all of the clock outputs that we can observe. So that suggests that they're central clock components, that they are important for all of the clock functions that we observe. And similar feedback loops are seen in animals, but the genes involved are different from each other. Any questions? All right. So I want to talk about using firefly luciferase as a reporter, as a tool to study this process. So have you guys heard about luciferase already? Yeah? Okay. So you know that this is an image of a firefly. It's a really useful reporter for us. And what's great about it is that it catalyzes this reaction where the substrate D-luciferin in the presence of ATP is converted to oxyluciferin and light is emitted. And it's great for a couple of reasons. Most importantly, you don't have to treat the organism with light to get that reaction to happen. So it's not like green fluorescent protein where you have to illuminate your sample to get light emitted. This is, these plants that express this transgene are able to glow in the dark from the enzyme that they're making already, and we spray them with the substrate luciferin. We don't force the plants to make luciferin, we provide it for them. So, I've already told you that light can reset the clock. So if we had to treat our plants with light to see this, then that would mess up the process we were trying to observe. So what we do is we take firefly luciferase and we put a normal plant promoter upstream of it. So there are many promoters in plants that can confer circadian regulation. So for example, the promoters of TOC1 and CCA1 and LHY, those all work really well. And we make this transgene and we inject it into plants in a technique I wasn't going to talk about. So then we have a transgenic plant here. And this is a really easy thing to do in Arabidopsis, much easier than in almost any other complex eukaryote. So it makes it, a really great model system. And so we end up with this. This is a transgenic Arabidopsis plant. It's emitting luminescence. And we take movies where we have a computer take a photo every 2 hours. And we can do that for a week. And so you get this kind of a pattern where the plant glows and then gets more dim and then glows again. So this is all false color. The blue is darker, less photons being emitted, and the white is brighter, more photons being emitted. It's just a little bit easier to visualize this way than if I did it as one color. And we can do this with about 1,000 plants per experiment, so we get pretty good throughput. And when we measure the luminescence given off by individual plants, we get this kind of a pattern. So these plants are grown in a constant environment, and yet you see peaks of luminescence occurring regularly, about every 25 hours in this case. So this is great for a lot of reasons. One, we can look at individual plants. We don't have to grind up and extract RNA from populations. It's non-destructive. If we find a mutant, we can go recover it and then get progeny from that plant. It's automated, so it doesn't require somebody actually being there every 2 hours to take a picture. Actually, was the setup when I was a postdoc. So we had teams of people where we would trade off, whose shift it was to go take pictures every 2 hours. That was kind of a drag. So having the computer control is great. And we use this, yeah.
So you actually do 1,000 plants at once?
We do, yeah.
This is like 1/1000th of a snippet that you're getting of the whole image?
Exactly, right. So I mentioned Arabidopsis plants are compact. So if we have a 10 by 10 centimeter plate, we can have about 80 plants in that one plate. That's we assay them when they're a week old, so they're still small. So that helps with the genetics quite a bit. All right, so we've used this as a screen for finding clock mutants. So this is an average of plants that had a wild-type clock. You can see this lovely 24-hour rhythm in bioluminescence. But the pink shows the average of some mutant plants where the clock runs much more quickly. And so that's a pretty nice phenotype. It's robust enough that you can use it as an assay to isolate the gene responsible for the phenotype. Oh, this is kind of hard to see. But the pink here, you guys probably remember from high school your Mendelian genetics. If you have a recessive trait and a heterozygous parent, if you look in the progeny, so we can, if a het crossed to a het, and you look at the progeny, 1/4 will be mutant, half will be heterozygous, and they'd look wild type, and then 1/4 will be wild type. So that's what you see here. These 1/4 here are homozygous mutant. These 3/4 of the population here are either wild type or heterozygous. And these blue guys that you have trouble seeing, those are the wild type controls. So kind of outside the scope of today's lecture, but we can then take these mutant plants down here and find the stretch of genome that they all have in common, which is the stretch of the genome that contains the mutated gene that caused that short period phenotype. And that's in fact how how the field isolated the TOC1 mutant, the CCA1 mutant, and so forth. And people have used this approach in fruit flies, and actually they've used it in mice as well, but those are pretty tough and expensive screens. Yeah. Okay, so any questions about using the luciferase as a reporter or doing genetic screens? No? All right, so this is a research project that a former postdoc in my lab carried out over a few years. And so we wanted to know, we started out from the question of what genes are clock regulated in plants. Are we finishing at 10 till?
You've got another 10 minutes.
Another 10 minutes? Okay. Okay. So what genes are clock regulated in plants? I indicated that lots of aspects of physiology are clock regulated. So what Mike, my former postdoc, did was a microarray experiment. So that's, I think, another really interesting topic, but also complicated, so I don't want to get into the details. But basically, a microarray is a way to determine what genes are being expressed in a sample at a particular time. But the power of it is you can look at all the genes in the genome at once. So if you've ever heard about northern blots or those kinds of techniques, there you're looking at one gene at a time, and it takes you about a week to do one experiment to figure out what one gene is doing. If you do a microarray experiment, it takes you longer to do the experiment, but you've looked at 25,000 genes by the time you're done. So actually the handicap with microarray experiments is the data analysis. You get huge amounts of data. I did one of these experiments when I was a postdoc, and I literally spent 1 year analyzing the data from one experiment. So it's just a huge amount. So that's when you really, this is when biology needs statistics, is when you start looking at 25, 30,000 things at once. At any rate, so what Mike did was he grew plants in light-dark cycles for 1 week. That was to make sure that all of the clocks and all of those plants were synchronized. And then he released them into constant light and temperature on day 8. And then harvested tissue samples every 4 hours over 2 days. And for every one of those time points, he isolated RNA, and each of those samples was hybridized to a gene chip. This is one of those devices that allows you to assay expression of essentially all the genes in the genome at once. And this is Mike, his self-portrait. He's sitting in front of his computer analyzing the data, and he spent quite a long time analyzing this experiment also. And he concluded that about 10% of the genes were clock regulated. So this is called a heat map. So red represents a time when a gene is highly expressed, blue a time when it has low expression, and this is time of day at the bottom. So white is subjective day, so it corresponds to these points here, and gray is subjective night, corresponds to those points there. And then different genes are on the y-axis. So if we just look at the top here, these genes, about 400 of them, show peak expression around subjective dawn. If we go across, you can see they have trough expression about 12 hours later. And then 12 hours after that, they have peak expression again. So we see 2 peaks and troughs over the 2 days of the experiment. And we identified these genes by taking all of the raw data and fitting it to cosine curves, cosine waves. And so if it fit well to the cosine wave with a 24-hour period, we said they were clock regulated. So 10% of genes may seem like a large number, but you see something similar if you look in fruit flies or rats. It's a large fraction of the genome is clock regulated. Okay, but we wanted to take this, so this is the diagram of all of those clock regulated genes. You can see some differences between them. Some are very low amplitude down here. This is expression level on the y-axis and time of day on the x. Some of them have high amplitude. A lot of them have really low amplitude. So our experiment didn't tell us anything about the biological significance of this. Does it matter if something varies by 20% every day and night? Maybe, maybe not. It probably matters if it varies by 5 or 10-fold. So that's one thing we could sort of use as a tool to start to separate things from each other. But what we then did was say, okay, well, what pathways show more clock regulation than you'd expect by chance? So, for example, if you look at genes involved in photosynthesis, about 90% of those are clock regulated. Okay, that seems like it's very highly statistically significant. And in fact, we know that photosynthesis is regulated by the clock, but we wanted to find new pathways. And so there's a plant hormone called Auxin, and we found that about a quarter of the genes in the Auxin signaling pathway were clock regulated. And so that seemed to us like it might mean there was a causal connection between the clock and Auxin signaling. And so Auxin is a plant hormone that's involved in tropic responses such as growth. This is a little cartoon based on Darwin's book. And he noted that these seedlings grow towards light. If you grow plants in a windowsill, they grow towards the window. And he, being Darwin, he didn't just say, oh, plants grow towards light. He said, well, I want to understand what's going on. And so he did some very simple but elegant experiments. He found if he chopped off the top of the seedling, it no longer grew towards the window. He said, oh, okay, there's something important there. So then he put he didn't chop off the top, but he put a tinfoil cap on top of the seedling. And he found that it no longer grew. So he said, okay, maybe there's something about the tip there. But if you put a clear cover on the tip, it grew appropriately. So the tip of the seedling is perceiving light. And if you put the collar further down, it didn't interfere with things. And then just by looking at the shape of the plant, he could infer that different growth was occurring down here. So light was being sensed at the tip of the plant, and a signal was being sent down to cause differential growth further down the plant. And so that signal turned out to be this hormone called Auxin. So I won't go through the details of this pathway. I'm running out of time. But I will say, so we suspected that the clock was regulating Auxin signaling. And so we made a luciferase reporter, that is sensitive to Auxin levels. So under conditions of low Auxin, these are now individual transgenic plants with the camera far away from the plants. And you can see a little bit of glow. If we spray the plants over here with Auxin, you get a big glow, okay, because they're sensing that Auxin and they're making more luciferase. So that showed that indeed we made the Auxin sensitive reporter. But we wanted to know what was going on if we didn't spray the plants with Auxin. And that's this movie here. Now these are individual seedlings. Auxin is made mostly at the shoot meristeme at the top of the plant, but you can see that Auxin is also being made to the luminescence of that Auxin. Let me do that one more time. Okay, so especially if you look at the apex here, you can see they're bright, they're dimmer, they're brighter, they're dimmer. And you can also see little pulses of light going down the stem of the plant. And if we, it gets kind of fun if we start spraying those plants with different auxins. We can then see how their development changes and how auxin responses change. These plants were sprayed with the natural Auxin. You can see that big burst of light. And then they start to make a lot of roots. Okay, so Auxin is a rooting hormone. People use it when they're trying to take cuttings and get roots to form from the cuttings. And if you take an herbicide that's just a very stable Auxin, it's called 2,4-D. This was actually a component of Agent Orange that was used to defoliate the jungles in Vietnam. You get this. You get these plants that, like, they're putting all their energy into making roots. And so we can monitor Auxin signaling in those plants. So I think that's pretty cool. Anyway, but this was the point of our experiment, is we wanted to measure luminescence in the DR5 plants that we didn't spray with Auxin. And we found that indeed there was a 24-hour rhythm to luminescence. And I hope you can see that if you look at the shoot apex. And we can quantify this, and we get this kind of a pattern with the 24-hour rhythm and glow occurring. All right, so I'm going to skip a few slides, just in the interest of time. And so, all right, so we found through some experiments, I don't have time to go through, that the clock is regulating plant responses both to the Auxin that the plant normally makes, but also the responses to Auxin that we could spray on the plants. And so you might say, well, why? Why would an organism want to do that? And so I have a little speculation. This is not proof by any means. But this is a movie of a sunflower plant growing outside, and it's a time-lapse video showing that the leaves of the plant are tracking the sun. And that's a differential growth process that's mediated by Auxin. Okay? So the sun's overhead, the plant leaves are spread out pretty flat so they can harvest as many photons as possible. And as the sun sets, you see the same thing. The leaves track the setting sun. Okay, but then at night, look, the leaves collapse down into this neutral. And you can see before dawn, these leaves are already facing towards the east, right? Before the sun's come up. So this plant is absolutely ready to harvest all the photons of that morning sunlight when the sun comes up. And so you can see there's got to be coordination of a lot of things going on here. Plant growth, the time of day, right? So it's being able to know, I mean, I say no, but the plants collapse down to neutral at night, and then they face towards the east. And also direction. This plant knows which direction to put its leaves in. And so I think it's probably important to use the clock to coordinate growth with external light-dark cycles. And my suspicion is that, this linking between the Auxin hormone pathway and the clock is probably not the exception. That's probably the rule for signaling pathways in general. Okay, so we need to thank our Firefly collaborator. And also, most of the work I showed you today was done by Mike Covington, a former postdoc in the lab. And we have various funding agencies that I should thank as well. Okay, thank you.
You guys have any questions?
So I know you mentioned you didn't really invest some time about this, but when you did the transformation for your Arabidopsis, is that still bacterial transformation that you do or not?
Yeah, so we use Agrobacterium, which is a natural plant pathogen, and it has a special piece of DNA that it just injects into the cell nucleus. So we use Agro. Yeah, works really well in Arabidopsis, much better than most other plant species.
Any other questions, guys? Well, I thought that was a fascinating presentation. Thank you so much.
Thank you.
Thank our speaker. And